Hello, we are the Kanha Lab, and our project is using ensemble learning algorithms to predict scaffold matrix attachment regions. Um,
Yeah, so a quick introduction to our project. Scaffold matrix attachment regions, or SMARs, are regions of the human genome which bind to the nuclear matrix with some known DNA motifs. SMARs act as hotspots for retroviral integration, a process that allows for retroviruses to integrate into the human genome. SMAR regions influence the epithelial mesenchymal transition, or EMT,
Okay.
which is a process key to wound healing and cancer metastasis. Issues with SMAR-related matrix binding proteins have led to lower survival rates in triple negative breast cancer and pancreatic cancer as well. Many methods have been developed to identify SMARs. However, prediction rates are reported to be unsatisfactory at this time.
Okay, so previous studies have compiled a list of DNA motifs, markers, and sequences of SMARs, but there's yet to be a definitive, accessible database of SMAR sequences in the human genome. So for our project, we used R Studio to create individual classification models based on data of SMARs from HeLa cells and motifs. We currently use three models: XGBoost, Random Forest, and K-nearest neighbors, which we will explain in this presentation. In the future, we plan to transition from individual classification models to an ensemble model, as well as finding more lab-based data that is accessible. And now we'll move on to our methodology. So first, we started with our literature review. From there, we branched out to achieve two main goals. One was data collection, which we will go into more in a later slide, and another was to identify SMAR motifs. For data collection, we found our data and then we compiled it and processed and cleaned it. And after identifying SMAR motifs and cleaning up our data, we started on our first model, which was the random forest model. After finishing our random forest model, we started to build other models, as well as started error analysis and model tuning. We are currently at the final stages of these models and the tuning. And in the future, we plan on creating more visualizations in addition to the ones that we already have, finishing our documentation for our R package, and publishing our R package with a paper, which we've already started working on.
So one of the first steps, as seen in, I guess, our mm, mm, methodology, was our data collection. So we were able to collect around 497 SMARs from the ENCODE project, which helped give us the SMAR start and end positions. And using AppScript code with Google Sheets and UCSC genome browsers, we were able to extract the full sequences of the DNA. Using this data, we were able to create a pipeline to process the data and search for motifs, and some examples specifically for the origin of replication are ATTA, ATTTA, and ATTTA. We also were able to analyze AT richness with the following sequences, and we chose AT richness in specific because a lot of the papers that we were reading about SMARs show that SMARs binding proteins are more prone to binding to AT-rich regions of a DNA sequence. We also started using or tried to use SMOTE as a technique for more data, and SMOTE is a technique to kind of generate like artificial DNA sequences.
Sorry about that. For our tools, we used R Studio as our main coding platform. We also used various R packages to help build our models. And then we used GitHub to store our code and our file history. We got the start and end numbers for the SMARs from each chromosome from the ENCODE project. And then we extracted the DNA sequence using the UCSC genome browser. We use Google Sheets to format and sort our data and get rid of any errors, which we later downloaded and put into R Studio.
So we've just started working on some simple UI of our tool that we hope to published in the future. But here you can see like just a simple interface where the user can upload a FASTA file and they're able to see their full DNA sequence as well as a header. And this header is usually from like wherever they downloaded their file from. So in this case, an example was that I downloaded like a FASTA DNA sequence from NCBI, and it just displays the whole DNA sequence as well as the header for it. So in the future, we hope to kind of integrate the current models that we have and maybe show a proper prediction of SMARS along with like a visual. Yeah. So, like one of the first models that we integrated was random forest, and how it works that it combines multiple decision trees to improve accuracy and uniqueness or robustness. So, once doing that, it creates multiple subsets of the original data set by randomly sampling with the replacement, and this process is called bootstrapping. Then, like a decision tree is made on each subset of the data with its own subset features. And I guess the overall output is calculated by taking the majority of the outputs from each decision tree. So why we kind of started with this model was that it uses multiple decision trees, and so it's less prone to overfitting, which is when a model almost memorizes the data that is given to it, and it doesn't result in accurate results. And we also noticed that it's less sensitive to outliers in our data. Um,
Okay, so, oh, sorry.
I'm so sorry. Yeah. So the overall accuracy that we got was around 88.8%, seen in the confusion matrix on the top right.
Okay, so now we're going to move on to our next model, which was the k nearest neighbors model. So how this model works is that it finds the k closest data points to the new target data points, and it computes the distance between the target data point and all the other data points around it, which are its neighbors. And then it predicts and then it finds the shortest distance, and then it predicts the class of the data point, which in our case would be SMAR or not a SMAR, based on majority vote with its closest neighbors. So some benefits of it is that it is easy to implement, and it is a good base model relative to other machine learning models to start with. And it's also backed by the IEEE paper that we use to review the models and select them. It also helps capture relationships, which in our case is about smarts. Some drawbacks is that it can be computationally expensive, but that's not really an issue with our project at this stage because our data set is on the smaller side. So if we look on the confusion matrix to the right, we can see that our model has a 67.9% accuracy rate here. And when we use K-fold cross-validation, the accuracy rate becomes 68.7%. And then we have our XGBoost model. So XGBoost is a gradient boosting algorithm uh, that is that combines the predictions of multiple decision trees together to get a final result. Some pros of it is that it does historically have a high accuracy rate, as stated in the IEEE paper that we used to select this model, and it also prevents overfitting, which Sathya explained before. Some cons of it are memory usage, but again, that's not a problem for us right now due to the small nature of our data set. When we used K-fold cross-validation, we got our accuracy rate to be 82.3%. And this model specifically was way more prone to bias because of the less negative data that we had initially. So we used the SMOTE technique to add more data, and then we are currently in the process of adding these additional sequences created with SMOTE to the other models. And the reason I mentioned this is because if you look at the confusion matrix on the left, you can see that the numbers used in these confusion matrix are way higher than the numbers for the other models' confusion matrices. And so, yeah, that's the reason why.
Yeah, so now on to our, I guess, general model metrics. We measure this using sensitivity, specificity, precision, recall, accuracy, and kappa. And so sensitivity is basically how well our models were able to identify positive cases. So true positive rate. Specificity is how well our model was able to identify n- n- negative cases. So true negative rate. Recall is the same thing as sensitivity almost, and precision was the fraction of values that were correctly predicted as positive. Accuracy was just our overall accuracy, and kappa it was the um, measurement of agreement between our observed accuracy and our expected value. And so overall, our like random forest model had an accuracy around 0.88. Canon had a 0.93, and our XGBoost had a 0.82.
And then in conclusion, our takeaways so far are that we've managed to achieve a relatively low error rate for the classification of SMARS in comparison to existing computational models, and that through our literature review, we've compiled a collection of genetic motifs that are indicative of the presence of SMARS regions. Some current limitations of our project include the need for additional lab-based data, both negative and positive, in order to further balance our dataset, and the bias that some of our current models are prone to, and also the identification of allele repeat over reliance, which is essentially telling us that in the future, we need to ensure that our model doesn't just recognize motifs, but instead is capable of making actual connections on what represents a SMAR, so that it doesn't simply work on a small subset of SMARS regions, as do many other computational models in the past. And then for future work, we're transitioning towards an ensemble model in order to improve our capabilities. We're working on publishing our paper and tool and finishing our user interface so that people can use the tool. And then the significance and the applications of our model are that it will help researchers carry out SMARS-related research, which means potential improvements in prognosis for patients with metastatic cancers thanks to new related discoveries, and also further identification of hotspots to retroviral integration, which could lead to better methods of treating retroviruses. Thank you. And we would like to thank our advisor, Mr. Khanna, for his support and guidance, and also ASDRP and the Olive Children Foundation for giving us this opportunity.